So, as we do traditionally here, we spend the first few minutes just talking about our careers and how we got to where we are. But then I'll move into the talk, which is about designing a cell transplantation therapy to repair spinal cord injury. And this is a big project, so I'm only going to be touching on some aspects of it. So if you've got questions about other parts, I'm happy to answer those after the seminar. So I started off in my science career doing agricultural science, and I did a Bachelor of Agricultural Science with honours at the University of Melbourne. And the focus of that degree was to look at scientific production of animals and food and fibre using the variable, unpredictable resources of Australia and to meet the market needs. So it's about using science with the end user in mind in context of markets and economics. So it's a very diverse and broad degree, but also in depth in other areas. After my undergraduate degree, I worked for the Rural Water Commission in Victoria, and I worked in Shepparton, which is in central Victoria, for a year, looking at how irrigation can be used to help agricultural production. I was then fortunate enough to be offered an industry scholarship to do a PhD, and I was actually paid more to do a PhD than I was to work for the Victorian government. So I took the option of doing the PhD, and I did work on fructan metabolism. That's carbohydrate metabolism in pasture grasses. In Victoria over summer, the grasses dry off and the sheep are left to graze brown grass. And the idea was that if we can increase the carbohydrate content of the grasses prior to senescence, then the sheep would get better quality feed over summer. Because typically sheep, they lose weight over summer because of the poor quality feed that they are faced with. And so I did a PhD in agricultural science at the University of Melbourne. And I also did a Bachelor of Letters in International Development, which was looking at government systems and economies of developing countries, because my ambition was to work overseas in agriculture. We worked with people from Africa and South America, and my dream was to work overseas. That didn't quite happen because at the end of my PhD, life got in the way of things, and I took what I thought would be a short-term job in neuroscience. And Brian Key at the University of Melbourne offered me a postdoc job looking at carbohydrates for axon guidance in the brain. So I worked on carbohydrates in grasses, I worked on carbohydrates in brain. It can't be too difficult, is it? The similarities here on the left, you can see a picture of grass, and here on the right, picture of glial cells. They have the same sort of shape. Apart from that, there's not much similarities. So it was certainly a big learning curve for me to jump into that area. But the reason I took it was because at that time, the olfactory system, the sense of smell, was being discovered in great detail. And the Nobel Prize was given to Buck and Axel for their work looking at the topographic map of the olfactory system. And it regenerates throughout life. And so it was sold to me that if we can work out how the olfactory system regenerates, then we might be able to apply that knowledge to other parts of the nervous system and enable therapies to be developed to repair the injured and diseased nervous system. And when we first started, that was just a great sort of line you could use for grants to get funding, because no one thought it'd be possible to regenerate the spinal cord or brain at that time. We're talking in the mid-1990s here. But that was the idea. If we can find out how we can use the olfactory system knowledge to repair other parts of the nervous system, we might get somewhere. And then I applied for a NHMRC Peter Doherty Fellowship, and this was a 4-year fellowship, and I was fortunate to get that. I had a good PhD. I got 7 publications out of it, 5 first author publications. And because I was different, because I came from a different background, I think that gave me an advantage. And so for our early career researchers, don't be frightened of jumping ship and going to a different area if you've got skills and aptitude that could be applied to the new area. And I did that at the University of Melbourne, and then Brian Key moved to the University of Queensland and I went with him. And then after that, I got 3 NHMRC project grants in a row. This is the time when success rates were quite high. I think it was about 33% to get an NHMRC grant. Since then, it's dropped down a lot. But because I kept on getting the grants and the fellowships, and then I stayed in this area and didn't end up going overseas that I'd originally intended. But I love the work, and I love the work because I've discovered things. And I think one of my advantages is that I haven't done formal neuroscience training at university, so I don't have the dogma. I don't know what to expect according to what the textbook says. And so when I look at things, I have my mind open to new ideas and new ways of looking at cells. What we're seeing here is a movie I made of nerves, axons within a living zebrafish. And so we labeled the zebrafish neurons with fluorescent tag, and you can see the growth cones. You can see the growth cones at the end here, and the filopodia, and they're searching through that environment. Now remember that here there's lots of living cells around it. There's fluid flow happening, and these axons and these filopodia have to reach out and look for their targets. And this fascinated me. And this was the time when live cell imaging was just coming into existence, because we had the ability to label neurons with fluorescent tags to see them, and then the microscopes to be able to visualize that as it was happening. Then I made this movie, and this was deep within the brain of a zebrafish. And hopefully it's working on your screens. It's moving slowly. And you can see at the top here, there's a growth cone, and then there's filopodia, and they're moving in 3 dimensions. And all around there are cells and fluid flow. I actually have a very good movie of the fluid flow that happens between cells in living animals. And it's much more rapid than you would think, and much more complex. So it's not like a static environment that these axons are growing through. It's a changing environment, and yet they have to find their targets in that. It's a very complex environment. And so I spend my time looking at these and thinking, how do they do it? And trying to work out ways of improving that. What is really exciting about this movie is that this is actually real time. It's not a time lapse. If you look at the time in the bottom, it's every 2 seconds is every 2 seconds. And when I first made this movie, my boss, Brian, came in and he looked at it and said, wow, it's a really good time lapse. And I said, no, it's not. It's actually real time, and we're capturing it as we were watching it. And when we presented this at the Australian Neuroscience Conference in the early 2000s, people actually didn't believe it. And they didn't believe it because they didn't think that axons could move so quickly. And one of the reasons was because people have been doing time lapse in a dish, with cells in a dish, and they tend to move much more slowly in a dish. In the body, where it's more complex, they move much faster. And then I realized that we were focusing on axons, and this is an example of our work, but this is what other people do too. So here we have labeled axons. Again, it was a fluorescent tag, so we can see them as they're growing. And over time, you can see that the axons are moving. And many people in the field have been applying growth factors to these axons to see what would attract them or repel them, and they were making interpretations based upon that. But what I did was looked at the whole picture using my holistic approach, which I learned from my agricultural days. And when you look at differential inference contrast imaging of the same field of view as above, you can see other cells in there, and these are actually glial cells. And when you overlay the glial cells with the axons, what you notice is that the axons are actually taking a ride and moving with those glial cells. So we then looked at more detail. We had labeled glial cells that are red, and here you can see that the growth cones and the filopodia of those axons, which are green, are hitching a ride on the glial cells. So for me, this was the light bulb moment that if we want to get axons to grow, we need to get the glial cells to do their job, because the axons are hitching a ride on the glial cells. That's not to say all axons can't grow without glial cells. Some can, but if you want the best growth, you have to get the glial cells to do their job. This and the previous images were in vitro. These ones are in vivo. So we can see here that this is an axon growing across inside a zebrafish, and you can see how it touches a cell here and other cells you can't see, but they have very close relations to cells, and so they have to grow in their environment. And this is, so we worked on olfactory and sheathing cells, which are the glial cells of the olfactory system. And this is what people used to have, a static image, but when we use time-lapse imaging, we can see that the olfactory and sheathing cells are moving very quickly. And they have lamellipodia, they're interacting, they're migrating, they're making connections, they're losing connections. So if we want to regenerate the nervous system, we have to understand how cells interact, make connections, find those connections, and then make sense of those connections. And olfactory and sheathing cells have fantastic properties. This is a glial cell, which is red, and it's got green bacteria, and the glial cell is taking up those bacteria, internalizing them, and you can see that those bacteria are being accumulated around the nucleus, and where that glial cell has been cleaned up. So these glial cells have very good properties for repair and regeneration. They can remove cell debris and bacteria from the injury site, and then they can help the axons to grow. So if we focus on the olfactory and sheathing cells, we might get the neurons to do their job. So this is where we move into spinal cord injury. And we're focused on the individual here. This is Perry Cross, who had an injury when he was 19 years old playing rugby, and he was injured in a tackle and became a ventilated quadriplegic. So if we look at him, we can see that he's in a wheelchair. We might think he can't move, but also we have to understand that he can't feel and he doesn't have control of his autonomic functions. So it's not just a matter of getting people to move, we also have to repair the sensory system and the autonomic system as well. And then we have to make a therapy that these people want to have. And this brings me to Griffith University. So Alan Mackay-Sim was fortunate to get his funding for the National Adult Stem Cell Centre in 2008. And I wrote to him and said, Alan, I'd love to work with you because of the work he'd done in humans in 2002, where he transplanted olfactory and sheathing cells into humans here at Griffith University in Queensland. And his work showed it was safe, and recently he's been recognised for that with the Australian of the Year award in 2017. So back in 2007, I moved to Griffith University with the idea of abandoning looking at axons and focusing our work on the glial cells, because to get the glial cells to do the job means that we can get the neurons to follow. This is an animation that we had made, which sort of shows how the therapy works. Oops, it didn't work. I'll go back there again. Let's see if I can click it. So it's quite simple in concept. We take a biopsy of the nasal cells, put them into a dish. In that dish, there's a whole bunch of different cells that we have to separate out and retain the target cells. Then we put those target cells in a three-dimensional construct and transplant them into the injured spinal cord, and that can be via injection or open transplantation. And then once within the spinal cord, they can do their job to repair the injury site and allow the nerve cells to regenerate. Sounds simple, it's much more complex than that. But the inspiration for our work comes from the many scientists around the world who've been working on this one. This was a 2012 study from Cambridge University. This dog here was naturally injured. You can see his back legs are paralysed. He had olfactory and sheathing cells transplanted into his spinal cord, and he could regain coordinated hind limb movement after 6 months. And then with more time for those nerve cells to make connections and the muscles to relearn the movements and support the weight, he could also bear his own weight. So it shows that the therapy is successful. But in this study, not every animal responded, so we need to make it better. Then in 2014, a Polish team joined up with Professor Jeffrey Reisman from University College London and showed that this therapy can work in humans. This man had been stabbed in the back with a knife and had a complete spinal cord injury lesion, apart from a small lateral branch. And this shows that this therapy can work. Again, in this study, not every person responded. There were 3 people, only one showed good recovery, and that his recovery is still incomplete. He's still got some way to go, but it can work. And at the time, it was hailed as being greater than man walking on the moon. And that's partly because think about it, sending astronauts to the moon is quite achievable. You know that the forces are that are required. You know the speed. You know how much fuel you need, and so you can achieve it from an engineering point of view. When we think about the spinal cord, we don't know much at all. We don't know what the injury site looks like for each individual. We don't know what cells that we need to transplant. We don't know the chemicals and molecules that are in there that change the signaling in each patient, in each injury site. So to achieve what Jeffrey Reisman and the Polish team did in humans was absolutely incredible. Whether we can repeat that in other humans remains to be seen. So it gives us the inspiration to keep on working on it. The other thing too is that when the Americans sent the spaceship to the moon, they put billions and billions of dollars into it and had a coordinated national approach over a decade. So if we had that much money put into spinal cord injury, who knows what we might achieve. And so Perry Cross, after that, met me in the courtyard of N75 and he said to me, James, when are we going to bring this therapy back to Australia and when are we going to make it happen here? And that was a real dramatic statement for me because I realized I was being a scientist, I was doing science, publishing papers, and then planning the next experiments. When Perry said that we had to deliver a therapy, I realized that my role as a scientist is much more than just doing the research, but is actually to take on that responsibility of delivering a therapy. And quite honest, it terrified me at that time, and still it's a pretty incredible sort of thought that we could progress to clinical trial. But we have responsibility of giving it our best shot. So let's see what we can achieve. So the question is, how long till we can get back to a clinical trial, and what do we need to get to a clinical trial? So in 2016, we were fortunate enough to get the funding from the Clem Jones Foundation to establish the Clem Jones Centre for Neurobiology and Stem Cell Research. And we work on 3 main areas, which is the spinal injury project, the peripheral nerve injury project, and neurodegeneration. But today we're just talking about the spinal injury project. And we've got funding from many partners who also share our vision for what we hope to achieve. And it's careful communication about the risks and the hopes that we can achieve, but also being realistic that it's a long way to go. And that's allowed us to bring in a fantastic team of people. It's absolutely the brains in the team who allow us to achieve what we can achieve. And this is an old photo, and some people have left and we've got some new people, but we have about 30 different people. And our policy is that we recruit people who are different from our existing team. So then you need to bring in new skills, new approaches, and new ideas. We're 50% female, 50% male, and we come from about 16 different countries. And we are humans too. We're people who have strengths and weaknesses, and we work with that to have a big, happy family who hopefully can work together as a team to achieve what we can't do individually. So for us to progress the therapy, we had to think about what we needed. And we came up with a strategy which has 5 components for an effective therapy. That was to improve the purification of the olfactory sheathing cells, because at the moment it's not a consistent, effective method. We wanted to use drugs to stimulate the olfactory sheathing cells to improve their activities. I won't be talking about those 2 aspects today. Then we had to put them into a 3D nerve bridge, because the current approaches are to inject a suspension of cells, and most cells don't survive the transplantation. Within 24 to 48 hours, 90% or more of those cells are dead. So if you're injecting a fairly low purity population of cells and most cells are dying, but you're still getting good recovery in some animals, then it suggests to us that if we can improve the cell survival, then we will get better outcomes. And then we have to think about the transplantation method, which is to transplant a three-dimensional cell construct. And then that's the easy part. The hard part is then getting the people who receive those cells to do long-term effective regeneration. So three-dimensional cell culturing, Rajavali in 2005 developed the naked liquid marble method, which is a ball of liquid encased in a hydrophobic powder that enables us to create a mini bioreactor. And this allowed us to generate the first scaffold-free cell constructs. But it wasn't suitable because they're too fragile, so we had to change the way we did the cell culturing. And this is where getting some bright minds in, such as Mo Chen here at the front, allows us to achieve that. And I should also point out that we have here Brett Monagle, who's a clinician we work with. He gives us the biopsies and understands the clinical need. We've got Matt Barton, who has done peripheral nerve research, and of course Jenny Ekberg here, who's got incredible in-depth knowledge of the physiology and neurobiology of the cells. And so having different people in the team allows us to create new approaches. And this team here, we recently received the NHMRC Marshall and Warren Award for the nerve bridge proposal that we had in our NHMRC Ideas Grant. But what Mo did, and again with the help of Marie Viau, looked at what we call naked liquid marbles. It's a new method of growing cells in 3 dimensions. And this is like a mini bioreactor, scaffold free, and we can recreate what a nerve looks like. This image here in the middle, the red cells are factory incision cells, and the blue nuclei around the outside just show where the fibroblasts are. It's a cross section of a nerve. Over here on the right is our recreated 3-dimensional structure with our factory incision cells in the middle and with fibroblasts around the outside. And that could be created within 48 hours. And it self-assembles. We don't have to do anything to it other than give the cells the right conditions in which to grow. And we've got a patent application submitted for this, this method. But then those previous 3-dimensional constructs and system were quite small. Mo pushed the boundaries and made larger ones. And so this one here is 3 millimeters long, and it's made of 1 million cells. And so it just shows that we can keep on pushing the boundaries and making bigger and bigger constructs. If you compare this size construct to many other 3-dimensional organoids that are produced using neural cells, this is very, very big. Most are typically 500 to 700 microns in diameter and end up with a necrotic core. Our system doesn't have that, and they remain healthy and viable over the long term. So the idea was that if we could transplant these 3-dimensional constructs into the spinal cord, it would be a source of cells that would, and the factory incision cells would migrate out into the injury site over time. And I made this quite large field of view here for demonstration purposes because they migrate out over about 1 day, and the distance they migrate is about 2 millimeters. So let's play it again. And so nerve cells tend to regrow at about 1 to 2 millimeters per day. So the migration here of the olfactory incision cells is consistent with what we know can occur in the human inside our body. So if we can replicate this within the injured spinal cord, hopefully we can create a pathway by which the axon can grow across that injury site. And so I'm going to show you here a movie that we have from our animal studies in which we transplanted the olfactory incision cells into the mouse. It's a ladder here, and the mouse will run along the ladder. And to start with, it's a normal, healthy mouse, and it can grip those ladder rungs and continue on its journey. We then induced a thoracic level 10 injury. The mouse is completely paralyzed in its hind limbs. And then we transplanted a 3-dimensional construct of those olfactory incision cells, and after 4 weeks, it had regained some motor function. It wasn't perfect, but it was a fantastic achievement. So we had to look at the integration of those cells. And this is the spinal cord here, and here's the lesion site, a full transection, and this is a 3-dimensional construct that's been transplanted into the spinal cord. And when we zoom in on regions where those cells are integrated, we can see these yellow cells here have integrated and grown across that injury site. But we were particularly keen on looking at the cell survival. So in the literature, cell survival is about 1 to 2% after a few days. When we injected our suspension of cells, we had about 2 to 3% of cell survival. When we transplanted the three-dimensional cell constructs, the spheroids, we were getting about 8% cell survival. And when we transplanted the cell constructs with the 3D constructs with some cell suspension, we were pushing that cell survival up to about 10%. And this is a conservative estimate. If we use if we extrapolate it, we probably might be getting up to about 15% cell survival. So that's about 15 times more than what can be achieved in the literature. So we're pretty happy with that, but still have some way to go. We then looked at how well those olfactory and sheathing cells can integrate within the injury site. Here we can see olfactory and sheathing cells. This is not the same image as here, but this is what would happen. Here's the injury. There's a clear gap. When we transplant cells into here, in some animals, we will get the red olfactory and sheathing cells with very nice morphology growing across that injury site. And in white are the olfactory axons that are extending across that injury site. So there's a very close relationship between the olfactory axons and the glial cells. And here's a movie showing the interaction of those axons. Here the OECs are yellow in the injury site. And then when we zoom in, this is a rendering of that immunohistochemistry staining and fluorescence staining. The yellow is the olfactory and sheathing cells, and the white are the axons. And as we cut away, we can see that for some of those axons, the olfactory and sheathing cells are wrapping around the outside of the axons. You can see that particularly over here and here. And so this is exactly what the olfactory and sheathing cells should be doing. They ensheathe the axons. And so that protects the axons and guides them over that injury site. And so we can see at the cellular level that we're getting very good integration of the olfactory and sheathing cells with very nice morphology, and that the axons are growing across those cells. Here's a similar view, but without the rendering. This is the actual fluorescence imaging that you can see, which gives a bit more sort of realistic detail. And you can see that there's very close relationship between the olfactory and sheathing cells, which are red in this case, and the axons, which are white. Oops, I'll just go back a bit. So one of the problems in the human, and this is where we need to talk to clinicians, is that there's a scar. And surgeons would not want to cut away the scar tissue because it's too risky in humans at this point. Our animal studies suggest that might be useful, but in humans, we do not want to make the injury worse. So if there's scar across that injury site, maybe such as, for example, here or here, and surgeons do not want to touch it, then how do we transplant our three-dimensional construct into a human and still achieve good results? Well, fortunately, our surgeries, while they're very, very good, thanks to the great skill of our surgeons who are doctors and vets, we don't always get it right. And in this case, the three-dimensional construct, which are the red cells here, was transplanted above the injury site, not into the injury site. But what happened was fantastic, because when we zoom in on this one, you can see red olfactory and sheathing cells and white axons are growing very nicely. So in this situation, we believe that the axons are growing up into that glial nerve bridge and across that injury site. And so we can bypass the scar. This tells us we can bypass the scar, we can put a construct over that scar, not into the scar, and hopefully then the axons from either side can grow across that injury site. But up to now, our scaffolds, our structures were quite small. Previously, we've shown that they were about 3 millimeters in size. So Mo, I kept pushing Mo. I kept saying, make it bigger, make it better. And one day we had a lab meeting, and he came back after the lab meeting with this construct here, which is made from human olfactory cells, and it is 3 centimeters long, 30 millimeters long. And while it took 6 hours to make, so previously we were doing 24 and 48 hours, it took about 6 hours. And mine was fantastic. I said, can you make it quicker? And he said, yeah, of course, because of the lab meeting, he couldn't go back and terminate the reaction earlier. So we can now make those nerve bridges in less than 3 hours using our special technologies. That allows us to make bigger nerves, and that means we can then treat not just spinal cord injury, but also large gap peripheral nerve injuries. And this is fantastic, but many nerves are branched or have several tracks together. So then can we make nerve bridges that are suitable for other types of injuries? And so the answer Mo came up with was yes, we can make a branched nerve bridge, and using our 3D printing technology, we can now make nerve bridges to suit pretty much any type of injury site. And you can see here as an example, a facial nerve where it could be branched. And can you imagine if you have paralysis of your face, then you lose expression. So it's really important that we get therapies that are relevant for the clinicians and the patients. Beyond that, we can make multiple branch nerves. In this case, it's got 4 branches that can be used, for example, such as here, or in the brachial plexus, where there's particularly complex branched nerves. So what we envisage is that we can now image the injury site using metal technology, produce a template of that injury site, the appropriate nerve construct, make our nerve bridge here, and then hopefully transplant it into the human to repair that injury site. I'm wary of time, so I'm almost finished. So as a reminder, we look at cell purification and drug therapies, which we haven't discussed today, but we've touched on some of the 3D nerve bridge technology and the possible methods for transplantation. But the critical thing is getting rehab correct. And I'll just spend a couple of minutes just talking about the rehab options here. In Australia at the moment, if you have a spinal cord injury, you go to intensive care, then the spinal injuries unit, for example, where you learn to live with your injury, hopefully regain some function, and then learn to return home and return to the community. But for many people, it doesn't give effective outcomes. 75% of people with spinal cord injury do not work. Most live in poverty, and there's poor outcomes. Overseas, there's lots of intensive rehabilitation therapies available, long-term intensive rehabilitation that are not available yet here in Australia. So we need to think about what rehab can achieve. This is our partner in Melbourne, The Next Step. It's a rehabilitation facility, a commercial facility, and this is an example of what can be achieved if we allow ourselves to be imaginative. This man here was paralysed, spinal cord injury, and was told when he was discharged that he was a complete paraplegic and he'd have to learn to live with that. And he believed the clinicians, and that they gave the best diagnosis at the time, and he believed the clinicians, and he learnt to live with complete paralysis. But then after 4 years or so, he decided, well, what if I did intensive rehab? So he went to The Next Step, and I visited there and seen their facilities, and it's pretty inspiring what they can achieve. But what you'll see from this man is absolutely astounding. And I say it won't happen for everyone, but it could happen. So you can see here he's having a therapy. This is what he could achieve, minimal movement. But over time, he regained incredible ability to move again. You can see he's shuffling here. He can support his weight. That's critical. Now he can step slowly and turn. This gives someone incredible independence from before being on a wheelchair. And here we go. He can sit down. This is again incredible achievement for someone. But then he got much more rapid recovery. He could walk. He could jump. He could have coordinated movements, and then eventually he could run. When we showed this video to the Federal Minister for the NDIS, Stuart Robert, he just said straight away, wow, this rehab here has just saved the Australian economy $1 million a year, because that's the cost of the Australian economy, $1 million per year per person with spinal cord injury. So if we can only achieve this in, say, 10% of people, we're still winning. From a human point of view, of course, we're winning, but from an economic point of view, it's fantastic too. So we need to have a good rehab program where we put cells in as well, and then have rehab, such as what you can see here. So we need to do long-term rehab. For that, we need a clinical trial, and for clinical trial, we then need an app so we can record people's progress, because it's not properly captured at the moment. So we had to make an app, and with Michael Todorovic and Matt Barton and the App Factory down at Gold Coast, we made an app suitable for spinal cord injury. So you can see here, I'm just giving you as an example of this is what we had to achieve. This is way outside our area of expertise, but we work with the people who can do it. And this app was produced in 3 months, which is a fantastic outcome. So now the clinicians, the people with spinal cord injury, the service providers can use this. It's a secure system that meets all the required medical data recording. So then comes it to where are we in terms of going to clinical trial? Our stakeholders want us to keep on pushing towards clinical trial, and it's a lot of hard work to get there. We're doing our best, and we're trying to put everything in place at the same time. But this is what we envisage. We envisage that people will do a period of sustained functional therapy prior to cell transplantation. We take a biopsy, purify the cells, make a nerve bridge transplanted back into the patient, and then, as I said, this is the easy bit, and then the hard bit is to get people to continue doing a rehabilitation program at home, at a specialised centre, and then at home for a long time. That could be 6 months, 12 months, 2 years. So it's a big program that would take community effort to make it achievable. Let's bring it back to the moon landing. Wouldn't it be fantastic in the future if we could have a headline like this? When will they walk in terms of people with spinal cord injury? And also, it wouldn't be fantastic if the kids could actually be at school, so they could come home again. Hopefully we'll be released from the COVID-19 restrictions soon and get our kids back to school where they should be. But this is what we want to achieve as a community, so hopefully we can do that into the future. And again, it's the team who can make it happen, and not just our team in the lab, but all our partners external to the university, but also within the university, Griffith Enterprise, Development and Alumni, and the Office for Research, who help us put our story in the right context and get our partners interested in the project. So I will finish it there. Thank you very much, and I will now stop the sharing, which I did, and I'll try to get chat function up. So, questions. All right, here's some questions. All right, can we have some questions coming through? Okay. Kathy says, I mentioned in your introduction that even small improvements in sensation may make a big difference. Okay, can I expand upon that? Okay, so it's very easy for us to think that motor function is the most important thing. Sensory function is really, really important. Because, for example, pressure sores is one of the main reasons people with spinal cord injury end up in hospital. Also, autonomic dysreflexia. That's when there's a misregulation of the feedback between a lower part of the body and the upper part of the body. And that can be caused by a simple thing such as a pressure point or a shoe tied up in the wrong way. If the person can feel that pressure without even being able to move it, at least they can say to their carer, I've got a pain in my leg, can you just alleviate that pain? That means then that the pressure sore won't develop, or the autonomic dysreflexia won't go out of control, and the person won't end up in hospital. Sensory is very important. Perry cannot, for example, feel when someone touches him. He would love to feel his family and friends give him a hug. He can only feel from the neck up. So human touch is really important. So that's why some of the examples there of why sensory is important. And also, if you want your muscles to be able to move, you're going to have to feel. If your foot is touching the ground or your hand is touching the cup, if you can't feel how strong that grip is, then you can't raise it to your mouth. Or if you can't feel it's hot or cold, you might burn yourself. So you need that both of that to make sense of that muscular movement and feedback. Is there any overlap with it, from Sally Ann, is there any overlap with the therapy approach you're working on and potential treatment of burns? We actually have an idea of using our cell construct production method for skin repair. And I've just talked to a dermatologist only last week about what they would like to see in a cell construct for burns as well. So, and then of course there's nervous system repair in that as well. So, yes, the answer to that one is yes. We just need to think about whether we can do it and who should do it. So, okay, more questions. Andrew Rayfield, were there any similarities between the growth of grasses or plants to the growth of neurons you found? Yeah, so the similarities, what I like to do is think of what it's like to be a cell. I spend my time thinking I'm a cell being transplanted into an injury site, and what do I need to do to grow and find those targets? And there's positive and negative cues. If I can understand what it's like to be a cell, then hopefully I can design therapies. Same with agriculture. Agriculture, you've got to think about the holistic approach. If you change one input, you're going to be changing some other aspect of that system. So we also have that in the spinal cord. If you're going to be introducing something, you're going to be having some other effect. And we've got to be trying to predict what those effects are. Okay, Alex Christino. Do you think that the glia glides the axons or the axons guide glia? So certainly with olfactory sheathing cells, they tend to migrate, and particularly in the olfactory system, they migrate ahead of axons. And that's been shown by several studies, including some of our own. So it's not the case for all axons, because we haven't studied all axons. But certainly what we think it's like I'll give an analogy of a motorway. If you're going to be repairing the M1 motorway, no matter how many cars, which are the neurons, you drive down that broken motorway, if the bridge is broken, for example, you won't get to the other side. The glial cells make the bridge. Once that bridge is repaired, then those neurons, the cars, can cross that bridge. So what we do see is that those filipodi can extend out beyond the glial cells, but pretty much, at least in 2 dimensions, the glial cells are ahead of the axons. Alex also asks, are exosomes important for communication? Yes, absolutely. And Mariam's actually got a great project. Mariam Murtaza's got a great project she's working on, which is not exosomes, but a variation of the extracellular environment and what happens there. So yes, we have to also think about the molecular signals that are occurring, the exosomes, the other cell compartments that are happening. It's very complex. Lynn asks, how can we do the 3D cell culture? Unfortunately, that's still confidential. We've got a new patent application that we're trying to submit for the new 3D stuff, but the smaller naked liquid marble work is published, and you can certainly see what that does. Desi says, how long did it take for that patient to go from complete paralysis to be able to run? So that person, from the next step, I believe it was about 6 to 9 months. It was very quick relative to his injury. That would not happen for the vast majority of people. But what it does tell us is that there are some people out there who will respond. And how can we predict which people are going to be like that? We don't know. And did they use new techniques? No, they used standard rehab physiotherapy. So they didn't use any fancy robotics or devices. David Lloyd at Griffith University is working on robotic devices and assistive devices. They add a dimension that can be very useful, but just using regular physiotherapy rehab programs was sufficient for that particular person to get recovery. And that's what they did in Poland as well for that person who got recovery. And so while he can only shuffle and ride a bike, that did take about 12 months. And then still over several years, he's getting improvement over time. Another question here. Can it be possible to have immunogenic reaction, other adverse reactions? Absolutely. So we need to be very careful that we don't make the injury worse. The good thing about the olfactory and sheathing cells is that they can be taken from the same patient, so the patient's own cells. Marion Mataza has also published a review looking at olfactory and sheathing cells, and in their natural state within the olfactory system, they rarely or maybe never have been reported to form cancer, unlike other glial cells. So they you can think about it, they are constantly being exposed to pathogens and environmental insults. It's their job to constantly help the olfactory system to regenerate. So they are hardwired to not go out of whack and cause cancer. They're constantly being turned over. So they are quite special compared to other glial cells. So we are hoping that with good quality cell preparation, that we won't get adverse reactions and outcomes from the transplantation, but we have to be careful there. Adam Mackay Sims' work in 2002 showed there weren't any adverse outcomes using good cell preparation. And last question is from Sandra. Are you looking at microRNAs and association with neurological function? No, we're not at this point. We are looking at lots of transcriptomics, proteins, and other things like that too, but it's an area that we can progress into. So, any more questions? Because if not, then we can finish up. So thank you for your attention and thank you for your questions. Kathy says, well done, James. Okay, I'll let you all get back to it. And I think, did it work? Did the movies work okay? Were you able to see those movies? Can you give some feedback about that? Yes, says Kathy, fantastic. So it's good to know for other people who might want to present that these were large movies too. I had the presentation was over 300 megabytes. So feel free to use a large presentation. Great. Thank you very much for your comments. Look forward to the next seminars. I think it worked quite well. And thanks, Maria, for preparing it. Okay. Bye-bye, everyone.
Thanks, everyone.
Thanks, Maria.
Thank you.